Welcome back to Bite Size MCAT. This will be your short lesson on enzyme kinetics and inhibitors. Enzyme inhibition and the ability to interpret Michaelis-Menten plots and Lineweaver-Burk plots is extremely high yield for the MCAT and for your biochemistry classes. In this video, we'll go over the basics to Michaelis-Menten kinetics, how this is transformed to a Lineweaver-Burk plot, and the three major forms of reversible inhibition: competitive, uncompetitive, and noncompetitive inhibition. So I want to begin with some broad definitions to prime this content for you. First off, Vmax is defined as the maximum velocity of an enzyme-catalyzed reaction when all active sites on enzymes are saturated. Think of this as every enzyme being active at the same time. That is the maximum speed or output that we have. And this is related to enzyme concentration because as we increase enzyme concentration, our Vmax can also increase. Km, on the other hand, is the Michaelis-Menten constant and is the substrate concentration at which the reaction velocity is 1/2 of Vmax. We will see how this looks on a Michaelis-Menten plot later. Km is related to enzyme affinity. So let's take a quick overview of an enzyme reaction. First, we have an enzyme denoted as E and a substrate denoted as S. When these react, we form an enzyme-substrate complex denoted as ES, which then forms our enzyme back with a product denoted as P. A few rate constants that we need to be aware of is K1, which is the enzyme and substrate reacting to form the enzyme-substrate complex. K-1, which is the reverse of this reaction and denotes the ES complex reversing back to the enzyme and substrate. And K2 is the formation of enzyme and product from the ES complex. Km, the value we just discussed, can also be calculated from these rate constants by this equation. Km equals K2 plus K-1 over K1. This could be thought of as the rate constants of the dissociation of ES over the association of the ES complex. Another important note is that Km is a measure of affinity. If our Km is high, that means the rates of dissociation are higher than the rates of association. As a result, this means that our enzyme has low affinity. On the other hand, a low Km means that our rates of dissociation are lower than the rates of association. This means that our enzyme has a high affinity. A high yield concept for the MCAT is that Km is inversely proportional to affinity. Let's now take a look at a Michaelis-Menten plot and apply what we've learned thus far. On the y-axis, we have reaction rate, and on the x-axis, we have substrate concentration. If we have some enzyme and slowly increase our substrate concentration, we get this Michaelis-Menten curve of reaction rate. If we see this asymptote at which the reaction rate sort of plateaus, we get our value for Vmax. Remember back, Vmax is the max velocity when all active sites are saturated. Also remember that this is related to enzyme concentration. Now watch this. If we take half of Vmax, about right here, and follow this down to the x-axis, we find Km. Remember back to our definition of Km? It is the substrate concentration when the reaction rate is 1/2 Vmax, and that's what we see right here. So, this is a Michaelis-Menten curve, but when we talk about enzyme inhibition, we want straight linear lines. So what we do is take the reciprocal of both reaction rate and substrate concentration, and this is how we form a Lineweaver-Burk plot. Now, the MCAT will never ask you to convert between one and the other, but it's important to see how these relate. So here we have a general Lineweaver-Burk plot. Let's look at some important points on this graph. So the x-intercept is going to be calculated by -1 over Km, and the y-intercept is calculated by 1 over Vmax. Now, the slope of the graph is calculated by Km over Vmax. So again, the Lineweaver-Burk plot is sometimes called a double reciprocal plot of the Michaelis-Menten curve. This is a useful transformation to linearize the data from the hyperbolic curve. Let's now switch over to talking about reversible enzyme inhibitors and their effect on Vmax, Km, and how to interpret these on graphs. First off, we have a graph showing an enzyme without inhibitor in this light green color, and an enzyme with a competitive inhibitor in this brown color. An important note about competitive inhibitors is that it specifically binds to the active site of free enzyme. We will learn later about some inhibitors that bind somewhere other than the active site. Let's see what occurs on this graph when a competitive inhibitor binds to the enzyme. Notice that the y-intercept does not change. How do you calculate the y-intercept? The y-intercept is calculated by 1 over Vmax. So if it's unchanged, that means the Vmax has not changed. This is because we could add more substrates that will outcompete the competitive inhibitor. As a result, the Vmax doesn't change if we add enough substrate. What about Km? We see that the x-intercept increases. How do you calculate the x-intercept? The x-intercept is calculated by -1 over Km. So if the x-intercept increased, that is due to the Km also increasing. Because of the negative sign, increasing Km leads to a less negative number, leading to an increase in the x-intercept. This is because more substrate is necessary to reach 1/2 Vmax in order to compete with the inhibitor. So, big overview. Competitive inhibitors bind to the active site on free enzyme. This leads to Km increasing while Vmax stays unchanged. Next, I want to take a look at uncompetitive inhibitors. Again, we have our general graph with both enzyme without inhibitor and enzyme with inhibitor. Uncompetitive inhibitors can only bind to the enzyme substrate complex after the enzyme has bound substrate. This is at some allosteric site away from the active site. Let's take a look at the graph. We see this special parallel line pattern, and this is because both Vmax and Km decrease. Vmax decreases because the inhibitor binding to the ES complex creates this inactive complex. This means that fewer enzyme substrate complexes are capable of reacting, reducing the effective concentration of enzymes. Km also decreases because the inhibitor binding to the enzyme leads to the ES complex being sort of locked and inactivated. This reduces dissociation, which correlates with an increased affinity and lower Km. So, overview. Uncompetitive inhibitors bind to a separate site on the ES complex only, and this leads to a lower Km and a lower Vmax, which generates these parallel lines on the Lineweaver-Burk plot. Our last type of reversible inhibition is noncompetitive inhibition, which is a type of mixed inhibition. The MCAT cares more specifically about noncompetitive inhibition, so that's what we'll be talking about here. Noncompetitive inhibitors bind equally to free enzyme and ES complex. Similar to an uncompetitive inhibitor, this binds to an allosteric site away from the binding site. Just a note, mixed inhibition binds to both free enzyme and ES complex, but not equally like in the case of noncompetitive inhibition. Let's see what happens to our kinetics now. Our Km stays unchanged because the inhibitor binds at an allosteric site away from the binding site, and therefore it does not change the enzyme's affinity. Our Vmax decreases because the inhibitor binding to free enzyme and ES complex inactivates them, reducing the total effective concentration of enzyme. So, overview. Non-competitive inhibitors bind to an allosteric site equally on both free enzyme and ES complex. This leads to Km staying unchanged and Vmax decreasing. So in this video, you should have learned what occurs to the enzyme kinetics of each type of reversible inhibition. In competitive inhibition, the inhibitor binds to the active site on the free enzyme, and Vmax is unchanged while Km increases. In uncompetitive inhibition, the inhibitor binds to a different site on the ES complex, and both Vmax and Km decrease, giving us a graph of parallel lines. In non-competitive inhibition, a special type of mixed inhibition, the inhibitor binds to an allosteric site on both free enzyme and ES complex equally. This results in Km not changing while Vmax decreases. I hope all of this helped, and I hope all of it made sense. Thanks for watching.